Okay, welcome to this special IBB seminar. It's really a pleasure to host Ricardo Cruz-Acuña, who's assistant professor of cancer engineering. That's a new position that was created at Columbia. Ricardo's here at Georgia Tech as an invited speaker for the BioE Day tomorrow. So if you're not planning to go, I encourage you to go. Should be a good event. And we were able to wrestle him to come early and give a seminar to IBB. So Ricardo really is an outstanding researcher. I'm very proud of my association with him. He received his bachelor's in chemical engineering from the University of Puerto Rico at Mayagüez. Then he came to Georgia Tech to get his Ph.D. in bioengineering, working in my lab. He did a phenomenal job on a new project, engineering synthetic matrices for iPS-derived organoids, and also established those as a delivery vehicle for transplantation. And that resulted in a highly cited paper, patent applications, more funding, more students being tortured in the lab with his project. So he really was a great researcher, but he was a great team member in the lab, and everybody was very happy for him when he moved on. So after Georgia Tech, he started as a postdoctoral fellow at Penn with Anil Rusti. But about a year and a half, Anil moved to Columbia. Ricardo tried that long-distance research relationship for a while. This is all during COVID. Very challenging, so he ended up moving to Columbia and then finish the postdoc and secure assistant professor tenure track position in the dental school, again in cancer engineering. He has gotten all sorts of awards. He was a T32 fellow. I like to point out that he got the NIH KO1, which is really a big, award. So I'm very happy for him. And I'll share one funny story of Ricardo. I have many funny stories with Ricardo, but the funny one was that, Ricardo, like I am from Puerto Rico, we don't like the cold. And the project that he worked on was a terrific collaboration with Jason Spence and Asma Nosrat at Michigan. And Ricardo spent a lot of time at Michigan doing the experiments. And, as a cheerleader, I said, it's not that bad. So he won one time in, I think it was like January. I said, Ricardo, don't worry, it's not going to be that bad. Just buy a parka, you'll be fine. And I never forget, he called me one morning while he was waiting for the bus. And I could hear the wind howling in the background. And he was so miserable. This is before the experiments turned out great, right? He was so miserable, and he's like, what am I doing here? And I'm like, just hang out. This is a good thing to do. You'll be fine. And he's like, yeah, you're sitting down in your office, in the warm Atlanta weather. But he persevered and did a great job. So, Ricardo, I'm very proud of everything that you've done and excited to have you here.
Thank you. Thank you. Yeah. I remember you told me, hang in there, you got this.
Yeah.
Thank you for the introduction. That's very kind. Thank you all for being here, for your time. The last time that I was in this room, it was during my PhD thesis defense. So if you see me nervous, it's all the PTSD. But anyways, yeah, I'm happy to be here. I'm going to be talking to you guys about my work, particularly a project that we published last year. That we're very proud of in the Journal of Clinical Investigation. But first I'll introduce a little bit of what we do in my lab. My lab, we like to call ourselves the Biomaterials and Tumorigenesis Lab. And our goal is to create engineered hydrogel platforms to reveal novel mechanisms of tumorigenesis that can be leveraged for therapeutic studies and personalized medicine applications. We're particularly interested in looking how the extracellular matrix properties as well as disease-relevant mutations contribute to tumorigenesis. So when you go from preneoplastic lesions to cancer. We utilize different in vitro and in vivo experiments to sort of elucidate what are the mechanisms that drive this sequence so we can target it and inhibit the progression of cancer. We have mainly work in the esophagus, esophageal adenocarcinoma, but now we're doing some additional work expanding to other tissues and other diseases as well. I'm gonna highlight some of that work towards the end of the talk. But primarily, I'm gonna focus on what we published last year on esophageal adenocarcinoma. As a little bit of an introduction, esophageal adenocarcinoma is one of the two major types of esophageal cancer. It is the most common type of esophageal cancer in the Western world, and that includes the United States. Unfortunately, patients that suffer of esophageal adenocarcinoma have very low chances of survival. In fact, less than 50% chances of survival within the 5-year span, and those chances significantly decrease as the disease progresses. So in our lab, we're very interested in understanding what makes this disease so aggressive, what makes this disease have so poor patient outcomes. And one of those factors that we know that contribute to cancer and cancer being so aggressive is the extracellular matrix properties. Ourselves, as well as others, have shown that the changes in the extracellular matrix properties modulate the hallmarks of cancer. So, changes such as matrix stiffening or changes in the ECM, like ECM remodeling, they initiate matrix-induced intercellular signaling pathways through self-surface receptors that mediate all the hallmarks of cancer from resistance to cell death all the way to metastasis. And in the case of esophageal adenocarcinoma, it is not an exception. In fact, here what I'm showing is RNA sequencing data that demonstrates that there's an increase in the genes that produce extracellular matrix components on esophageal adenocarcinoma patient samples as compared to the normal counterparts. Similar here, there's some of our own data that shows that when we stain slides from a biopsy of a patient that suffered EAC and compare it to a normal esophagus, we see that there's a significant increase in ECM components like fibronectin and hyaluronic acid, which what it tells you is that there are changes in the ECM, not only changes in the composition, but also indirectly changes in stiffness too, because when you have more components in the same space, you're increasing also the pressure on that space. And then others have shown that these changes in the extracellular matrix properties have an implication on the progression of the disease, reduced drug delivery, and resistance to therapy. Now, the problem is that there are no current in vitro or in vivo models where we can dissect the contributions or the individual contributions of the ECM property. So how do we know what are the contributions of the changes in the composition of the ECM or the changes in the stiffness of the ECM, independent from each other, right? And that's basically what we're trying to focus on this project, to create such model in vitro in the lab that we can use to better understand independent contributions of ECM properties on the progression of esophageal adenocarcinoma. Now, to do that, we need a cellular system. And the cellular system that we decided to use was patient-derived tumor organoids. For those of you that are not very familiar with patient-derived tumor organoids, they are derived from patient biopsy. We get the biopsy from the clinic. We process that biopsy into a single-cell suspension. Then we grow in on a dish, on matrigel, on a 3D matrix. And those cells would then proliferate and form these organoids or three-dimensional structures that resemble the phenotypical characteristics of the primary tumor. And others have shown that they also resemble their transcriptional signatures, their genome, et cetera. So they're fantastic models to study cancer biology and drug response. We have shown in previous publications that when we generate organoid lines from different patients, and they're shown here in different colors, and we treat those organoids with different therapies that are available in the clinic, therapies that are more exploratory, these organoids can respond to therapy differentially, so in different ways. Some respond, some do not respond. But what's more striking about this is that we show that when we compare the response of these patient-derived organoids to the response of the patients that they were derived from, had in the clinic to the same drugs or treatment, they correlate 100%. So these EAC patient-derived organoids, they serve as avatars, or can serve as avatars to predict patient response in the clinic to treatment. Now, as for those that work with organoids, you probably know about Matrigel and how Matrigel is mostly used to it's sort of the most common material used to grow three-dimensional organoids, whether they're iPSC-derived or patient-derived or animal-derived. Now, one of the main disadvantages of Matrigel is that it doesn't allow you to modulate its physical-chemical properties in an independent manner. And I'll just give you an example of what I mean by that. If I want to reduce the stiffness of Matrigel, I can dilute Matrigel from like 100% to 50%. I can just dilute it with media. And of course, as expected, the stiffness of the Matrigel will be significantly less on those 50% as compared to 100%. Now, when I grow patient-derived organoids, such as the esophageal adenocarcinoma organoids, they grow differentially. There are significant differences in their size, as well as the number of organoids that are generated between the 50% versus 100% Matrigel, shown here in images as well. And also, they show different expression of tumor-associated genes as well. Now, the problem is that we cannot say that these changes that we see on their size, the number of organoids, as well as their gene expression, is because of matrix stiffness, because at the same time that we are diluting the Matrigel to reduce the stiffness, we're also diluting or decreasing the polymer density and the adhesive ligand density. So we are changing the biochemical properties, at the same time we're changing the biophysical properties, and then we cannot really attribute these observations to either of these properties. So to overcome this limitation of Matrigel, we decided to use for the purpose of our project an engineered hydrogel, given that engineered hydrogels are an evolving and important component of 3D organoculture systems. As Andres mentioned, myself did some work in his lab studying epithelial morphogenesis. Others have done similar work using hydrogels and organoids to study high-throughput drug testing. And others have done some work also studying tumor progression, particularly in liver and breast. So for purposes of our work, we decided to use a hyaluronic acid-based hydrogel, or NORHA. This was in collaboration with Jason Burdick. Back then, he was just at Penn. He moved to Colorado now. The reason why we decided to work with this material was because hyaluronic acid, as I showed in one of the earlier slides, is one of the major components of the tumor microenvironment. So the nature of this hydrogel is very relevant to the disease as well as to the model as well. This engineered hydrogel is based on, as I mentioned, hyaluronic acid, but it also has an RGD peptide, an adhesive peptide, RGD, arginine glycine aspartic acid. It's a component, or it's a short amino acid sequence that is found in different ECM components like fibronectin, vitronectin. And the hydrogel is also cross-linked using an MMP degradable cross-linker to allow for cell-directed remodeling of their immediate microenvironment. This is this gel is cross-linked through a photo-initiating reaction. We're using blue light, and I can talk about the different advantages of using this hydrogel system, but the one that is most relevant to the project is the fact that we are able to control its stiffness as a function of cross-linker density and independent of the amount of hyaluronic acid or RGD peptide. So what I'm trying to say with this is that we can we can control the biophysical properties of the material, like matrix stiffness, without changing the amount of hyaluronic acid or RGD peptide, but without changing the biochemical properties of the material. So what we believe is that this would allow us to study how matrix stiffness modulation can activate esophageal adenocarcinoma-associated genes in our patient-derived organoids. therefore revealing novel underlying mechanisms of the disease that can be leveraged for therapeutic studies. So first things first, we wanted to make sure that the organoids were able to grow in our hydrogels. So we embedded these patient-derived organoids in soft hydrogels. Is about 100 pascals, very similar to those to the stiffness of matrigel, storage modulus of matrigel. The organoids were pretty happy within the hydrogels compared to matrigel, so the viability was pretty much the same. The organoids were also able to show markers of differentiation, as well as EAC-associated proteins like mucin 5AC and cytokeratin 8 to similar levels as they did in matrigel. So this data is important because we're showing that we can grow these patient-derived organoids in an engineered hydrogel, that we know exactly what it is made of, and that we can then further control its properties and see how they affect organoid formation and differentiation, et cetera. So in that sense, the next set of experiments that we did is that we decided to grow these organoids in hydrogels of different stiffnesses. So we went from 100 pascal, I remind you that's very similar to matrigel, to 1,000 pascals. So 1,000 pascals now falls within the range of what's been reported as the stiffness of the tumor microenvironment. So this range of stiffnesses is relevant to the disease as well as to our model. And as you can see from the graphs as well as from the images, the organoids that were embedded in the stiffer hydrogel had a significant, they grew larger in size as well as in number as compared to the organoids that were grown in the softer microenvironment. And this is important because it actually reflects what's been observed in the clinic, which is that cancer thrives in a stiffer microenvironment. So we're able to model that in vitro. Now, the next question that we had is, what is modulating this phenotype? So how are these cells translating these mechanical signals into intracellular signaling pathways that are, causing this phenotype? So, after we did some digging in the literature, we stumbled upon the YAP-SOX9 axis, which in the context of esophageal adenocarcinoma, it's been shown that it endows stem-like properties to the cancer cells. Now, it is unknown what activates this transcription factor in esophageal adenocarcinoma. So we looked into it in our samples and in our experiments, and we saw that the transcriptional level, both YAP and SOX9 were significantly increased in the organoids that were embedded in the stiffer hydrogel as compared to the other softer hydrogels and matrigel 2. When we looked at the protein level, we also saw a significant increase in the expression of YAP and SOX9 in the organoids embedded in the stiffer hydrogels as compared to the soft hydrogel. So this to us was very, we were very happy about these results because the importance of this data is that we're able to provide an explanation to the field, which is that perhaps the way this cancer works and the way YAP and SOX9 worked within this cancer is due to the activation of the YAP-SOX9 axis by matrix stiffness. Next, we looked into cell proliferation as stained by Ki67. We saw that there was a significant increase in the amount of proliferative cells within the organoids that were embedded in the stiffer hydrogel as compared to the softer hydrogel. We also looked at CD44. CD44 is a marker of cancer stem cells. And we also saw a significant increase in its expression in the organoids that were embedded in the stiffer microenvironment. So what we decided to, or what we thought that this data was suggesting is that matrix stiffness is mediating this stem-like properties on these cancer organoids through the activation of YAP and SOX9. Now, in order to sort of validate that hypothesis, we did a set of experiments to prove that But before I got into that, we also did some in vitro work, or sorry, some in vivo work to sort of complement the observations that we saw in vitro. Now, for the in vivo works that we decided to do, we wanted to do a xenograft transplantation experiments. It's a very common type of experiments in cancer research. Basically, you embed organoids into the back of the mice xenograft, but in our case, we did a little twist where we embedded the organoids in the hydrogels, the stiff and the soft hydrogel, and stiff or the soft hydrogel, and then we implanted them in the back of the mice for a month. And then after that amount of time, we collected the implants, and we looked into organoid growth, proliferation, et cetera. And as you can see here, there was a significant increase in the size and as well as number of organoids that were embedded, that grew within the stiffer hydrogel as compared to the soft hydrogel in the in vivo environment. We also looked at the organoid differentiation. They were able to differentiate within the hydrogels in the in vivo environment. And we looked at the YAP expression, and then we see, similar to what we saw in vitro, we saw a significant increase, not only the expression of YAP, but also in the nuclear localization, which means it's been activated, as well as SOX9 expression too. We also saw a significant increase in the proliferation of the cells within the organoids and the stiffer microenvironment as compared to the softer microenvironment. So in general, we were able to validate some of the observations that we saw in the in vitro experiments, but in this case, in this in vivo environment, further supporting a hypothesis that matrix stiffness modulates organoid growth, differentiation through activation of YAP. In this in vitro and in vivo settings. Now, to further validate this hypothesis and also take the project into a more translational approach or aspect, we decided to do some therapeutic studies. So again, if the hypothesis is that matrix stiffness is modulating tumor organoid growth and proliferation and stem-like properties through YAP, that means that we inhibit YAP, we will be reverting the effect of matrix stiffness within this organoid. So if that is the case, that means that if we decide to use berdaporfin, which is a small molecule inhibitor of YAP, in our in vitro systems, we will see an inhibition of the effect of matrix stiffness. And that's basically what we saw. So here I'm showing some representative images of the organoids embedded in the stiffer hydrogel. Those organoids, they had a significant, they had a high expression of YAP and nuclear localization. Now, but when you compare those organoids to the group that were treated with berdaporfin or the small molecule inhibitor YAP, we see a significant reduction in their size as well as YAP expression as expected. And that reduction is very similar to the organoids that were grown in the softer material, regardless of treatment. Therefore, validating our hypothesis that matrix stiffness is mediating this phenotype on the organoids through YAP. Now, we wanted to look into it also in our in vivo setting as well. So for that, we did a similar experiment to what I explained before. We embedded organoids in a stiff hydrogel versus soft hydrogel, and then we implanted them in the back of the mice, a xenograft. Then we treated the mice with berdaporfin, the small molecule inhibitor of YAP, for a month. And then after that time, we took the implants, and then we looked into organoid formation, growth, proliferation, et cetera. And to our pleasant surprise, we did see that the organoids that were embedded in the stiffer hydrogel in the control mice, they had a lot of organoids, big organoids, and then when we would compare those to the organoids embedded in the stiff hydrogel, but on the treated mice, there was a significant decrease on their size as well as in their numbers, and then again, that level of size and number is very comparable to the organoids that were embedded in the softer material, regardless of treatment. Of course, we had to check into YAP expression to sort of validate our hypothesis. We see that there was a significant reduction in YAP and SOX9 expression in the organoids embedded in the stiff hydrogel on the mice that were treated with diporphin as compared to the control group. We also saw a reduction in the proliferation of cells in the organoids, as well as the expression of CD44. And again, this reduction in the expression of these proteins was very similar to the organoids, similar to the expression of the organoids that were embedded in the softer material, regardless of treatment to the mice. So, in conclusion, I hope I've convinced you that our NORHA hydrogels was able to support our patient-derived organoid viability, growth, and proliferation. Our data suggests that there was a matrix stiffness-dependent effect on the growth of these organoids and the activation of the YAP-SOX9 axis that would, as a result, endow stem-like properties on these cancer cells. And also that we were able to establish an engineered 3D platform that can potentially be used to test matrix stiffness-activated therapeutic targets using small molecule inhibitors. In our case, as an example, we use verteporfin to inhibit YAP. Now, a little bit of as I teased you guys before, or next things that we're doing in our lab, going back to sort of my initial diagram, we're interested in understanding how changes in the extracellular matrix properties, as well as disease-relevant mutations, contribute to the metaplasia-dysplasia carcinoma sequence, or basically going from pre-neoplastic lesions to neoplasia. To do that, we're doing some work studying Barrett's esophagus. Barrett's esophagus is a precursor lesion of esophageal adenocarcinoma, or also known as intestinal metaplasia of the esophagus. For that, we're doing a similar approach where we are generating Barrett's esophagus patient-derived organoids in our lab. We have already started to do some initial experiments, initial studies that are very, very interesting. Again, we are looking into how matrix stiffness potentially can push that metaplastic state on these organoids to a more dysplastic state. So from pre-neoplastic to a little more towards neoplasia. So we can better understand what are the contributions of matrix stiffness on tumor genesis. For that, again, we're using this hydrogel system where we can control its stiffness as a function of cross-linker density. We have done some preliminary studies where we embed these organoids in hydrogels of different stiffnesses, and similar to what we saw with esophageal adenocarcinoma, with this Barrett's esophagus organoids, we also see there's significant increase on the density or the amount of organoids that are generated within the hydrogels, as well as their size, and also we see some crypt-like architecture happening on these organoids that are more resembling, this more dysplastic state on the organoids as compared to those embedded in the softer gel. We also did some single-cell RNA sequencing on these organoids, and we were able to identify significantly upregulated genes that are involved in esophageal adenocarcinoma. So there are genes that are involved in cancer, and we saw them in these pre-cancer organoids as embedded in the stiffer hydrogel as compared to softer hydrogel. So this data definitely suggests that there's an implication, or that the matrix stiffness has a role on the neoplastic progression of this Barrett's esophagus organoids. So we're doing some further studies, applying some in vivo models as well. We're also working in collaboration with Jim Wells with some iPS-derived esophageal organotypic raft cultures. In this case, we're interested in studying how p53 mutations contribute to the neoplastic progression of intestinal metaplasia of the esophagus. So it's a pretty cool model of being able to adapt to our lab, and hope we can do some further studies with that model. And also we're now also expanding to oral squamous cell carcinoma, collaboration with Chang Lee at the College of Dental Medicine at Columbia. We are utilizing nanoindentation to measure tissue stiffness from patient biopsies that are on tissue slides that we get from pathology. So we're trying to use slides from pathology, which also that are readily available, to study how matrix stiffness on the tissue throughout disease progression. And we've been able to see, here I'm only showing basically healthy versus cancerous, but we also have some older stages in between, like dysplasia, and we see that there's a progressive increase in matrix stiffness, or tissue stiffness, I should say, and that is, as a function of disease progression. So we're also implementing some of these organoid models to study oral squamous cell carcinoma, and then we're excited about what's going to happen with that type of work as well. And with that, there's a lot of people that I had to thank, but I highlighted the people that had a direct impact on the work that I've shown. Our lab is small, but it's very productive. We're actually getting a fourth person this fall. We're excited about it. I have I guess I had to thank a lot of collaborators and a lot of supportive mentors that I have at Columbia and also here at Georgia Tech as well. Funding sources, and with that, I'd like to thank you all for your attention. I'm be happy to take any questions.
Thank you, Ricardo. I'll ask one question before Elijah jumps in. Actually, two questions. So, do you need RGD on the HA matrix?
Good question. We do. So we tried with our DG, and then the organisms do well. They don't survive. So they grow, but they don't grow as much, significantly less.
And then the other question I'm more interested is sort of the memory and the reprogramming in the system. So if you grow your organoids in soft and stiff, take them out, put them in the mouse without the matrix, do they still form the tumors, or they have the stiff matrix needs to be present there throughout the in vivo experiment?
That's a very good question. We did not try that, but we did try it in vitro. We grow organoids in soft and stiff hydrogel, and then we took the organoids from the stiff hydrogel, and then we passaged them into another stiff and also soft. And then we saw that the changes were those, even if they were cultured in the stiff hydrogel, when we passed them to soft hydrogel, they did not grow as much, they did not proliferate as much. So there's some reversibility on the effects that this environment has on the organoids. Yeah.
Yeah, great talk. I was interested in your RNA-seq data. I was wondering, I was expecting to see YAP present in one of those transcripts. Did you just show a couple of the genes? Are there more that are upregulated?
So you mean for the bulk of the presentation? Yeah. So we did RNA-seq data. So YAP was there. It was not, and in the RNA-seq, it was not significantly upregulated as we did see in qPCR, but the most important thing, based on literature, is that we saw those changes at the protein level, because really the way YAP as a transcription factor, the way it works is that it, at least in cancer, it's not that it's necessarily being there's a lot of more transcription happening. It's really more that at the protein level that it's not the way it works, that when it gets activated, it doesn't get degraded on the cell. So it accumulates on the cytoplasm, and then it translocates to the nucleus and binds to SOX9 and other downstream targets. So really the changes that we see, at least in esophageal carcinoma, in YAP expression activation is at the protein level, so not necessarily at the transcriptional level. Does that make sense?
Yeah, that makes sense. And one more quick question. Have you thought of also looking at FAC expression too and FAC phosphorylation? I know it's been shown in other cancer biopsies that you see, increased FAC and increased YAP. Have you thought about that? Have you done that?
Yeah, we haven't, but yeah, you're right. FAC has been shown, I think, for hepatocellular carcinoma, to have a big role there, but we haven't looked into it carefully. Yeah.
Other questions?
I know, sure.
It's on.
There we go.
Hi. Yeah, thank you so much for flying out to give us this great talk.
Thank you.
I was curious, so with the YAP data that you found, were you able to show correlation with what you found in vitro with what's found in like those same patients with their kind of building off like the RNA sequencing data set? Like, have you seen those kind of correlations in patient biopsies?
Yeah, so we haven't made, for this project, we haven't done direct comparisons. The organoid lines that we're working with, we work with multiple for this project. They were part of a biobank that we had. Those patients have passed away by now. But what we have seen is that the observations that we made here do correlate with what clinicians do see in the clinic, which is that in the case of esophageal carcinoma and other cancers too, they do see an increase in tissue stiffness, even in the patient's esophagus as the disease progresses. But we haven't been able to do like a direct comparison with patient organoid as we did what you're alluding to, the data that I showed some of the earlier slides. That was a separate story that we published in 2021. There we did do the comparison, but that was not in the context of matrix stiffness. So I can't really talk about that particularly. But
Yeah, that was actually like my follow-up question. So with those earlier studies, was that a different class of small molecule drugs that were used to perturb the system, and that also was able to recapitulate what you saw in patients?
Yeah, exactly. So yeah, it was yeah. There were different drugs. Some of them, some of them are readily available in the clinic, and there was one of them that was more exploratory, but they were not targeting YAP. So yeah, that was to be honest, that was a really cool project in the sense that it shows how translatable these models can be. So now we're also doing some studies on the side where we're trying to look precisely into that, can we compare patient data with what we see in vitro to matrix stiffness is also involved in therapy resistance. So if we try some of these drugs that are available in the clinic and in our in vitro system, can they recapitulate resistance to therapy for those patients that do show resistance to therapy versus the ones that don't? So there's a lot of questions that can be answered, and I think with this model that we have generated, I think we can tackle some of those questions, and we're excited about that.
Gotcha. Thank you.
Yeah.
Hey, great talk. Have you tried deriving your PDOs in the HA hydrogel? Because I'd imagine like you might get a different population of cells than the ones that you get from deriving them in matrigel.
You said driving?
Like deriving them?
Deriving them.
Yeah. Establishing the
sorry.
models. Yeah.
Yeah, deriving the patient-derived organoids on the hydrogel, bypassing matrigel, basically. Yes, we have tried that. We have been as successful as we have with matrigel, which is not 100%. Unfortunately, it's hard to get patient samples, particularly from this type of cancer, because the population that we serve in the Heights in New York City, we don't see a lot of cases. Good for them. We don't get a lot of cases for this. But the ones that, the few that we've gotten, we'll try to generate on the hydrogels. We've been able to establish the lines in the lab. Some have failed. But the same thing we see with matrigel. Some do well, some don't. It depends on every patient is different, so every organoid line is different.
Did you try them in like both matrigel and your hydrogel? And like, are they different at all? Yeah.
Yeah, we've tried that, and the ones that we've been successful with hydrogel, we've also been successful with matrigel. So I can't say that which one's better than the other. Yeah.
You should have tried PEG-MAL, but
That's actually gonna be my question. Ricardo, great talk.
Thank you.
I wanted to know what were the decisions behind selecting the NORHA material for your studies, and if you considered using a synthetic material, since that was your entire PhD was working with synthetic material versus the NORHA. And the other question that I had was related to the YAP inhibitor that you use. Do you envision that being translated into like a therapeutic agent for the clinic, or what are the clinical applications of that?
Yeah, both great questions. So the reason why we decided to go for NORHA as opposed to Pefformal, nothing wrong with Pefformal, but everything's great. Oh, that's true. That's true. No, it's mainly because hyaluronic acid is a major component of the tumor microenvironment, particularly in EAC. So we wanted to use a bio-inspired gel. It was more from for relevance to the disease, and also it's easier to tell the story too. So that was the main motivation. Plus the collaboration that I had, that was with Jason Burdick, that was his gel. So, and then the second question was, so verteporfin has been in clinical trials, not for esophageal adenocarcinoma. I think it was for liver cancer, but I'm guessing now. And it did not. It was not. It didn't work out. But it's a proof of concept. And what we hope to show is that with this engineered hydrogel system, we're able to use as a platform to try different drugs. And our next steps, we hope to try drugs that are perhaps currently on clinical trials, or that are currently in the clinic, so that we can continue to expand on the feasibility and how good this platform can be for drug testing. Yeah.
I'm coming back.
Yeah, just one more quick question. Have you ever thought of looking at that transition from healthy tissue to cancerous tissue, and maybe also looking at senescence too? Because I've heard that in the field of like cancer versus senescence. And is that possible with this system, or no?
Well, in terms of healthy to cancer, no. We've only been working with diseased, like some sort of like Barrett's esophagus, adenocarcinoma, or so preneoplastic lesions, but they're not cancer, but they're still lesions. Growing healthy patient-derived organoids are really hard, very difficult, even on matrigel. So that's something that it's even from a technical perspective, it's kind of a little bit, it's difficult to study. But so our focus have mainly been on disease organoids. And then you asked about senescence. You mean like on the cancer organoids or the healthy?
I sort of think of the transition between healthy cancer, maybe healthy and senescence.
Yeah. So I guess in that we only work with disease organoids, and we haven't compared normal versus disease in our system. Yeah.
Hi, thank you. I had another hydrogel question, similar to before.
Say a lot, it was not clear.
Yeah.
Hi, Garcia.
Yeah. So with that upregulation of hyaluronic acid in this cancer, you're probably also going to see, since hyaluronic acid is a humectant, an increase in interstitial pressure in that interstitial fluid. So is that something that you can decouple in your hydrogel model when you're increasing cross-linking density? How does that impact
Decoupling, sorry, the stiffness of what?
So stiffness and like swelling, or that interstitial pressure. Is that something that you can decouple in your model? Is that something of interest?
Great question. Nobody has ever asked me that. I don't think so. I don't see how in this system, to be honest with you. That's very interesting. Something that we've been considering is looking into viscoelasticity, but then that's something we cannot do with this model either. We will have to change the hydrogel system. But yeah, no, I guess the answer is no. Thank you.
What is the natural tumor microenvironment like in relation to matrix stiffness? Because I understand the idea of engineered matrices and looking into matrix stiffness, but I would want to see how it relates to the actual environment of the cancer actually in the body.
Yeah, that's a very good question. So the stiffness of the tumor microenvironment it's a wide range. It obviously depends on the tissue, it depends on the stage. So what's been reported is that tumor stiffness goes from, in storage modulus, can go from 1,000 pascals to 5,000 or more. The hydrogels that we're working with, the stiffer ones were 1,000 pascals on purpose because we wanted to enter that range of tumor stiffness being reported, so that the stiffness is relevant to the disease. Yeah.
So Michelle, probably the best study on that was Val Weaver's paper on Cell, and what they did is they're looking at breast cancer cells. And in the mouse model that they used, they showed that if they added inhibitors of lysyl oxidase, which crossings and stiffens the tissue, they had less aggressive forms of the tumor on the same model. So that's probably the best in vivo comparison of the role of stiffness with tumorigenesis.
Yeah, there's not a lot out there, fortunately, on that. But it's just difficult to measure tissue stiffness in patients. But from what we were able to research, it was a wide range, and we were able to get those cells to be within that range. Yeah.
Okay. Well, if nothing else, please thank me. Join me in thanking Ricardo. Great job. Thank you so much.